Additional psychophysical tests of the V1 Saliency Hypothesis. Part 2. A fingerprint of V1 cells tuned to conjunctions of features. Let's have a visual search task in which human observers search for a bar that's unique. It is an odd one out. In this example, the target bar is unique in color. All other bars are identical to each other. Let's say this observer has a reaction time of 500 milliseconds to report this odd bar. For example, the task is to report by a button press whether this bar is in the left or right half of a display filled with such bars. Here is another example. Now the odd bar is unique in orientation. All other bars are roughly the same as in the first example above. Let's say that this time the reaction time is about 600 milliseconds for this example. Now let's have the third example in which the odd bar is unique in both color and orientation, all else being the same. Could we now predict what the reaction time should be? Maybe around 400 milliseconds? Maybe around 500 milliseconds? Vish can predict this third reaction time from the two reaction times above, and this predicted reaction time depends on whether V1 has neurons tuned to both color and orientation. Let's recall that Vish states that the highest V1 response to each visual location represents the saliency or bidding value for exogenous attentional selection at that location. Let's apply Vish to this example. Imagine a simple case when V1 has just neurons tuned to color only and neurons tuned to orientation only and has no other types of neurons. Let these be the firing activity levels of neurons tuned to orientation responding to these bars. And all of them are tuned to roughly the same orientation, namely the orientation of these bars, evoking their responses. They have low response levels since they mutually suppress each other by iso-orientation suppression. Let these be the responses from the color-tuned neurons. All these neurons are tuned to the blue color of the background bar, except this one, tuned to the red color of the unique bar to excite it. And it has a higher response because it is the only one escaping the isocolor suppression of the other color-tuned neurons onto each other. This is the maximum V1 neural response to this odd bar. And according to our hypothesis, this response relative to the background responses determines the saliency of this target location. Let's denote this highest response as X. For example, it could be 100 spikes per second. The reaction time to report the target depends on this saliency signal X. According to Vish, the higher this V1 response signal, the shorter should be the reaction time. Of course, the transformation from V1 signals to reaction times involves multiple intermediate steps, such as starting from the V1 signals bidding for attention. These signals are read out, let's say by the superior colliculus, to identify the winning bid. And then the command is sent to the motor regions so that this observer shifts his or her attention to the unique bar and presses a button to report it. All these transformation steps can vary quantitatively from one observer to another. For example, some observers may have stronger muscles to give faster motor responses. But this qualitatively monotonic decreasing nature should hold for all observers, even though the exact shape of this curve can vary from one observer to another. Now we can do the analogous analysis for this example when the target bar is unique in orientation rather than color. Now all the color-tuned neurons responding to these bars will give suppressed responses by isocolor suppression to each other because all bars have the same color and all responding neurons should be preferring this same color. But among the orientation-tuned neurons responding to these bars, the neuron responding to the target escapes the iso-orientation suppression because it prefers the target's orientation that is very different from the background orientations. And this neuron's response determines the target's saliency, and let's say that its response level is Y. For the same observer, we could use the same transformation, and so this response level Y gives a reaction time, and in this example, it should be 600 milliseconds. Let's compare this current case, which gives a 600 millisecond reaction time, for example, with the previous case when the target was unique in color. The reaction time was 500 milliseconds, for example, with a winning bit response level X. Here, the winning bit response level is Y. So therefore, by this monotonic transformation, X must be larger than Y to give a shorter reaction time. We use subscripts to denote the unique feature of the target, C for unique color and O for unique orientation. Now we are ready to examine this case. The target is unique in both color and orientation, and we call it a double feature singleton. These are the responses of the color-tuned cells. The highest response among them is X, from this cell which escapes the isocolor suppression. And these are the responses from the orientation-tuned cells, and the highest response among them is Y, from this cell which escapes the iso-orientation suppression. So therefore, the winning bit is the larger one between these two responses, X and Y. Whichever cell responding to the target is more activated, its response dictates the saliency of the target. And then by this monotonic transformation, the reaction time to this double feature singleton target should be equal to the minimum of the reaction times for the corresponding single feature singletons. Therefore, applying our prediction here, given these reaction times, VISH predicts a reaction time of 500 milliseconds. We remind ourselves that this prediction comes from assuming a simplistic V1 without any neurons tuned to both color and orientation. Usually, reaction times are not fixed to a single value. Across different trials, the winning V1 response for the target fluctuates. In one trial, it may be 100 spikes per second. In another trial, it is higher or lower. And this should cause fluctuations in the reaction times, which has a distribution over trials. Similarly, we can have another distribution of reaction times for the orientation singletons. Then, this VISH prediction enables us to predict a distribution of reaction times for the double feature singletons using a Monte Carlo simulation. This is done by randomly sampling a reaction time from this distribution for the color singletons, and take another random sample here for the orientation singletons, plug these two samples into this formula, and this gives you one sample in this distribution. And repeat this process, you can get a whole distribution of reaction times for the double feature singletons predicted from reaction time data from the single feature singletons. Here is an example set of experimental data on these distributions from an observer for the two single feature singletons. We can then predict the distribution for the double feature singleton and compare this prediction with the actual distribution for this double feature singleton measured in the same experiment. Here we show the predicted and observed distributions on the same plot to make a better comparison. The blue and red curves are for the predicted and observed quantities, respectively. On average, the observed reaction times are shorter than the predicted ones. And this is also the case in data from a few more observers. It turns out that this is because our prediction assumes a V1 that is too simplistic, such that the assumed V1 does not have any neurons tuned to both color and orientation. So let's come back to our drawing board for the double feature singletons. We consider that real V1 has neurons tuned to both color and orientation. We have responses from neurons tuned to color only, and the largest response among them is X to the target, and responses from neurons tuned to orientation only. The largest response among them is Y to the target. Now we add a third group of neurons tuned to color orientation conjunctions, and let this neuron type's response to the target be denoted as Z. Then the maximum response to the target is the maximum of X, Y, and Z. And this could be larger than the original maximum just over X and Y only. Then, according to our theory, this perhaps a higher bit should lead to perhaps a shorter reaction time. Therefore, the original prediction should be revised by this one. Essentially, this equal sign is replaced by a less than and equal sign. And this quantity is called the race model prediction. Therefore, in real V1, the reaction times for the color orientation double feature singleton may be shorter than predicted by a race model from reaction times for the corresponding single feature singletons. This explains such data in some of the observers because this blue curve is from the race model prediction. In summary, with RTC for the color singleton and RTO for the orientation singleton, then for the color and orientation double feature singleton, the reaction times are likely shorter than predicted by the race model. This inequality rather than an equality is because V1 has neurons tuned to color and orientation simultaneously. Now we can apply the same argument to the case when the color feature is replaced by a motion direction feature. In this case, all bars have the same orientation, just like the case to the left. An arrow on each bar visualizes the direction of motion of the bar, so therefore this bar is the singleton. It is unique in its motion direction only, moving to the left, while all the other bars are moving to the right. Now let RTM be the reaction time to find this singleton. So here the subscript M denotes that the unique feature is in motion direction. Here is when the singleton is unique in orientation only, and all bars move in the same direction. And the reaction time to this singleton is RTO. Now this is when the singleton is unique in both motion direction and orientation. The reaction time for this double feature singleton is also likely shorter than that predicted by the corresponding race model. And now because V1 also has neurons tuned to both motion direction and orientation. Note that these two inequalities are the same, except that the subscript C for the color feature is replaced by the subscript M for the motion direction feature. So here we have a triplet of singleton cases involving features color and orientation, a second triplet involving features motion and orientation, and now we can extend to a third triplet involving features color and motion direction. So this is for a singleton unique in color only, all bars move in the same direction. And this is for a singleton unique in motion direction only, all bars have the same color. And this is for the corresponding double feature singleton unique in both color and motion direction. However, the reaction times for this double feature singleton are predicted to be the same as that by the corresponding race model, because V1 has few neurons tuned to both color and motion direction. So if we plot reaction times versus the three types of double features for the double feature singletons, and if the race model predicts these kind of RT values, then VISH predicted RTs should be shorter than the race model predicted one when the double feature is CO, color and orientation. And also when the double feature is MO, motion and orientation. But the VISH predicted RT should be equal to the race model prediction when the double feature is CM, color and motion. This is a signature of V1 since it uses the V1 property that V1 has neurons to feature conjunctions CO and MO, but not CM. However, a signature of higher cortical areas, such as V2, will be like this. All the reaction times should be shorter than the race model predicted ones, because the higher cortical areas have neurons tuned to each of these three kinds of feature conjunctions. The experimental data supports the prediction by the V1 signature, and this suggests that higher cortical areas are not involved in visual saliency, at least for these kind of visual input stimuli.